Hello everyone, how are you all? After a long time, I am going to give a lecture on phenomenon of infection. The infection chain of plant pathogen consists of three stages, that is survival, dispersal, and third one is the infection process. So the infection process is the third link in the infection chain after survival and dispersal of inoculum. What is infection process? Infection process is nothing but establishment of pathogen in the host plant. Establishment means entry and colonization of pathogen in the host tissues. And inoculum. Inoculum is the infective propagules coming in contact with the host plant. So whatever infective propagules which can come in contact with the host plant and can initiate the process of infection, that is known as inoculum. And inoculum potential, it is the ability of that inoculum can produce the disease. So here we will see the definition of that inoculum potential. It is defined as the resultant of action of environment, vigor of the pathogen to establish an infection, susceptibility of the host and amount of inoculum present. This definition was given by Diamond and Horsfall during the year 1960. Or it also defined as energy of growth of a parasite available for infection of a host at the surface of the host organ to be infected. This definition was given by Garrett during the year 1960. These two definitions we can follow for the inoculum potential. If you will take the specialized pathogens like rust and powdery mildews, in such cases very few or even one spore can be capable of causing infection successfully. But whereas non-specialized pathogens like Pythium, Phytophthora, Rhizoctonia and Sclerotium, here in non-specialized pathogens high density of inoculum is required for producing the infection. So in case of specialized pathogens, very few or even single spore can be capable of causing infection, whereas in case of non-specialized pathogens like Pythium, Phytophthora, Rhizoctonia and Sclerotium, they will require the high density of inoculum on the surface to produce the infection. Coming to the different factors affecting the infection process. So these factors can be divided into three categories. First one is the host factors, second pathogen factors, and third environmental factors. So now let us discuss each one by one. Coming to the host factors, first one is the susceptibility of the host. So this susceptibility nature of the particular host, it can be controlled by DNA and it is an inheritable character that transmitted from parents to the next offsprings. This is the susceptibility nature of the host. And second one, disease proneness of the host. Disease proneness of the host, it is decided by the external factors like host nutrition. If you are applying more nitrogen, then it makes the host more susceptible, and if you are having more potash application, that leads to less susceptibility. So this disease proneness, it can be determined only with the external factors. Coming to the pathogen factors, first one is the virulence or aggressiveness of the pathogen. Aggressiveness of the pathogen means it determined by genetic material which is inheritable. Like in the host factor, susceptibility nature of the host, here virulence or aggressiveness of the pathogen, it also determined by genetic material of that particular pathogen. High multiplication rate of the pathogen, that is chances of infection increases with high rate of multiplication. If the pathogen is having high multiplication rate, then there is a lot of chance for the infection process. And high birth rate and low death rate, these are highly essential for establishing a successful infection in the host plant. And coming to the third factor, that is proper inoculum potential. Whatever inoculum is available for causing infection, it should have a particular potential to create the successful infection within the host plant. Suppose already I told in the previous slide that in case of specialized pathogens like rust and powdery mildew, even very few or even one spore is capable of causing infection successfully. In case of non-specialized pathogens like Pythium, Phytophthora, Rhizoctonia, their high density of the inoculum is required for those pathogens on the surface of susceptible host for creating the successful infection. These three belongs to the pathogen factors, that is virulence or aggressiveness of the pathogen, high multiplication rate of the pathogen, and proper inoculum potential. And coming to the third factor which influence the infection process is the environmental factors. Environmental factors also play very important role for establishment of successful infection by a pathogen. So like temperature, what is the relative humidity available during the infection process, and moisture available on the host plant, and these are very important for survival and dissemination and infection process. So these environmental factors, they will determine the three processes like survival, dissemination and infection process. Total infection chain depends on all these environmental factors. Next, coming to the stages of infection process. Let us see what are the different stages in the infection process. There are mainly three stages. First one is the pre-penetration stage, penetration stage and post-penetration stage. So pathogen will execute some stages for giving successful infection, like before pre-penetration what it will do, and in penetration stage what pathogen is going to do, and post-penetration what will going to happen. So each one we are going to discuss elaborately. Coming to the pre-penetration process, in pre-penetration, depending upon the plant pathogen activity, these plant pathogens are classified into two categories. First one is the active invaders and second one is the passive invaders. So active invaders means they will make an aggressive effort to gain entry into the host cells. That means they will execute some force on the host plant to make its entry. Whereas passive invaders means they will not execute any aggressive effort. In active invaders, they do not require help of any external agencies to gain entry into the host cells. So they can enter themselves without taking the help from the external agencies. But in case of passive invaders, they require the help of external agencies like insect vectors, wounds that caused by the agricultural implements. Examples for the active invaders are phytopathogenic fungi and phanerogamic parasites. And in case of passive invaders, plant viruses and phytopathogenic bacteria. Suppose if we'll take the plant viruses they are particulate in nature and they do not have any capacity to enter into the host cell. So what they will do, they do not make any aggressive effort for entry, but they will depend on different insect vectors like thrips, mites, white flies. Through these insect vectors, they will make entry into the host cell. And in case of plant bacteria, and bacteria they do not have any dormant structures, so there will be no pre-penetration activity except just they will multiply in the infection drops on the natural openings like stomata, hydrathodes. So they will do multiplication activity to make the successful entry. And in case of plant nematodes, nematodes show only some orientation towards root surface before exhibiting actual penetration. In case of fungal pathogens, pre-penetration includes like spore germination and growth of the germ tube that resulting from this germination, it will be exhibited on the host plant. And germination process, that means it is activating from the low metabolic activity to high metabolic activity. So this is essentially a change from low metabolic rate to high metabolic rate, and it involves a change from near dormancy to intensive activity. So for this, energy sources are needed like carbohydrates or fat reserves. Whatever present in the propagule, they will utilize these energy sources and these spores will start germination. Fungal invasion, that is chiefly by germ tube or structures derived from them, like whatever structures derived from the germ tube, they will help in penetration of the particular host plant. Like if you will take some fungi, Rhizoctonia solani, Armillaria, milia. The hyphae, it can act in concentrated way to achieve the penetration. Suppose if you will take the Rhizoctonia solani, when the fungus coming in contact with the root surfaces, first it forms the infection cushions and aprisoria. Here we can see that aprisoria formation in case of Rhizoctonia solani, and from this aprisoria multiple infections takes place by means of infection pegs. Here it will produce the infection peg, and from this infection peg it will produce the hastoria to absorb the nutrients from the individual cell of the leaf surface. Okay, next coming to the example rhizomorphs. In case of Armillaria melia, the fungus hyphae, it will form the rhizomorphs on the infected tissues. That is, rhizomorph is nothing but root-like mycelium which forms like root-like structures. That is aggregation of hyphae into rope-like strands. And only these can cause the infection. That means in Armillaria melia, these rhizomorphs can produce the infection. And coming to the second one, penetration. Now I discussed about the pre-penetration activities. So what are the different pathogens exhibit different pre-penetration activities. Now coming to the second stage, penetration. Here pathogens penetrate the plant surfaces by either direct penetration or indirectly. Indirect penetration means either through wounds or natural openings. So these wounds and natural openings, which pathogen can be used, that is bacteria. Bacteria, it will enter into the plants mostly through the wounds and less frequently through the natural openings. So maximum bacteria will enter into the host plant through the wounds, that wounds made by the agricultural implements or insect vectors like that. And in case of viruses, viroids, molecules and fastidious bacteria, they will also enter through the wounds. These wounds made by the insect vectors, or while feeding the insect vector, it will make the entry of the virus into the host plant. In case of fungi and the nematodes and some parasitic higher plants like Cuscuta, they will enter through direct penetration and less frequently through natural openings and wounds. Means they will execute some aggressive force to make their penetration into the host plant. First, now let us discuss about the indirect penetration, then followed by direct penetration. Indirect penetration means through the wounds and natural openings. So let us see what are the different organisms they will make their entry into in a indirect way. First one is the wounds. These wounds caused by like farm operations or hailstorms or insect punctures, etc. They will help in the entry of different plant pathogens into the host cells. So organisms which cause storage diseases, if we'll take some storage diseases or ripe rots, they will enter mainly through the wounds, and these wounds caused by the farm operations. Examples, if we will see, at least we have to remember one or two examples for writing in the exams. So examples, if we will see for the pathogen which can enter through the wounds are Rhizopus, Gliosporium, Aspergillus, Penicillium. These Aspergillus and Penicillium, these used to cause ripe rots, and these used to cause storage diseases in the fruit crops, and Colletotrichum, Diplodia, etc. So these are the examples which can enter through the wounds. And then another example is Macrophomina phaseolina, which used to cause charcoal rot and seedling blight diseases. And another pathogen causing brown rot of fruits, that is brown rot of fruits caused by Sclerotinia fructicola. It enters through the wounds that caused by insect punctures. And another one is the Dutch elm disease. This Dutch elm disease is caused by Ceratostomella almi or Ophiostoma nova almi. This Dutch elm disease enters through the wounds. These wounds caused by elm bark beetle, that is Hyalurogopinus and rufipis. And coming to the second one, wounds already we covered, and now natural openings like stomata, uh, then hydrathodes, and through stomata here there will be a variation in the behavior of germ tube of particular pathogen at the time of penetration through the stomata. because every germ tube will exhibit a different behavior. If we will take some examples like Puccinia graminis triticale, here the iridospore when it germinate, then it will form a germ tube, and germ tube on approaching stomata, then this germ tube swells at the tip to form a appressorium. After formation of appressorium in the stomatal aperture, then from that appressorium a blade like wedge which grows through the stomatal slits. Here you can see a blade like wedge which grows through the stomatal slits, and it swells inside to form a substomatal vesicle. From this substomatal vesicle, Haustoria used to be produced into the individual cells to absorb the nutrients from the individual cell of the host. So this is in case of Puccinia graminis triticale. That means it used to produce a substomatal vesicle. From that substomatal vesicle, Haustoria will be produced. And another example is Perinospora destructor, which produces the disease onion downy mildew. Here the germ tube continues to grow after the formation of first appressorium. Even appressorium formation completed, but germ tube continues to grow in case of this onion downy mildew. And another example, Pseudoperinospora cubensis, that causes the disease downy mildew on cucurbits. Here this pathogen, what it will do is, hyphae first penetrates the stomatal aperture and it will swell to form a substomatal vesicle, like in Puccinia where appressorium it produce the blade like structure, and substomatal vesicle how they will form. Here Pseudoperinospora cubensis also, it that hyphae first penetrates into the stomatal aperture, then it will swells to form a substomatal vesicle. From that substomatal vesicle, it produces the hyphae again, which grow to form the Haustoria in the adjacent cells of the leaves. And another example, Mycospirilla musicola, which used to cause Sigatoka leaf disease on banana. This Mycospirilla forms a small structure, what we call as stomatopodium, over the pore of that stoma. Means over the stomata pore, it will produce the structure, that is stomatopodium, after growing for few days on the surface of the leaf. Then after producing the stomatopodium, a hyphae arises from it, from the stomatopodium, and it grows into the substomatal chamber and swells to form a vesicle, which in turn give rise to a hyphae. That hyphae penetrates into the palisade tissues. Another example, this is the bacterial example, Xanthomonas campestris pathover malvacearum, which used to cause black arm of cotton, Xanthomonas fascioli, which causes bacterial leaf spot of green gram, and Phytophthora infestans late blight of potato, Albuga candida, which causes white rust of crucifers. These are the different pathogens, and iridospores of Puccinia graminis triticale. All these pathogens, they will enter into the host plant through stomata. Coming to the second natural opening, that is lenticels. Lenticels are the raised pores. They will permit the exchange of gases between the environment and internal tissue space of the organs. These lenticels generally we can find on potato also. Through these lenticels, some pathogen they will make their entry into the host plants. Example, Sclerotinia fructicola, which used to cause brown rot of fruits, and Streptomyces scabies, that is scab of potato, and Phytophthora ericae, which used to cause Mahaly disease of areca nut. And third natural opening, that is hydatodes. Hydatodes are the open pores which used to secrete water in the, they used to present in the epidermis or leaf margins, typically at the tip of marginal tooth or serration. Here you can see the hydatodes which used to be present at the margins. Through this water used to come out. Through these hydatodes, Xanthomonas campestris pathover campestris, which used to cause the black rot of crucifers, this pathogen will make entry through these hydatodes. Coming to the direct penetration, now we covered indirect penetration, that is indirect penetration happening through the wounds, that wounds caused by the agricultural implements or insect vectors, and natural openings like stomata, lenticels and hydatodes. Now this is the direct penetration. Direct penetration, if direct penetration has to happen, means the pathogen has to execute some force on the host plant. So which pathogen will execute force means, especially we know that fungal pathogens and nematodes. Fungi, nematodes and higher parasitic higher plants which are capable of penetrating the host surface are directly. And plant generally, if they have to penetrate directly, means they have to overcome some barriers which are already present on the host plants. That plants are provided with different mechanisms of defense, which include that structural features of the host, presence of chemical coverings on the cell walls, and anti-infection biochemical nature of that protoplasm. So whenever the pathogen coming in contact with the host plant also used to have its protective barriers. So automatically this pathogen has to overcome all these barriers and it has to establish infection for its successful multiplication and to proceed for further generations. So hence the pathogen should have mechanisms to overcome these barriers for the direct penetration. So what are the different barriers existing on the host plant? That is, first the pathogen has to break the physical barriers, chemical barriers, or it has to enter through non-cutinized surfaces and through cutinized surfaces. First let us discuss about the breakdown of physical barriers. How they will break the physical barriers. So viruses, they have no physical force or enzymatic system on their own to overcome these structural or chemical barriers of the host. So how they will enter? When they come in contact with the host protoplasm only through wounds. Then bacteria also, they are mostly weak parasites and they cannot employ any force to affect the penetration. And fungi and nematodes, these are the only group of plant pathogens that employ force for direct penetration of the host plant. And fungi penetrate the host plant directly through a fine hypha that is produced directly by the spore or mycelium, or through a penetration peg that is produced from the aprisorium. So fungi can enter into the host plant directly through either through hypha produced from the spore or mycelium, or through the penetration peg produced by the aprisorium. And these structures, like these physical barriers, they will exert on a pressure on the surface, that which results in stretching of the epidermis, which becomes thin. Then the infection peg punctures it and affects its entry. These mycelium or penetration peg, when they are produced by the fungi, what they will do, they will exert some pressure on the leaf surface, so that this epidermis will be stretched. After stretching of this epidermis, that it will become very thin in nature, so that when these fungal structures can enter the leaf tissue very easily. And coming to the second barrier, that is breakdown of physical barriers. So host is provided with different defense mechanisms against invasion of that pathogen, like presence of cuticular layer on the epidermis, that is cuticle layer, which used to be present on the epidermis, and lack of suitable nutrients for the pathogen in the host cell to grow, and presence of inhibitory or toxic substances in the host cell which can inhibit the pathogen penetration and pathogen entry, and exudation of some substances which are toxic to the pathogen, or they are stimulatory or antagonistic to the particular pathogen. These are the chemical barriers. If we'll take some examples, glands in leaf hairs of Bengal gram, that is chickpea, Caesar areatinum. In these leaf hairs, they used to contain one chemical substance, that is malic acid, that is antifungal in nature, and it will provide resistance to the infection by rust fungus, that is Euromyces caesaris areatini. This rust fungus is unable to infect chickpea because of that leaf hairs which containing the malic acid. If you take the red color onion, which used to contain red scales, that red scales contain a chemical compounds protocatichoic acid and catechol, These substances will provide protection to the onions against the onion smudge pathogen, that is Colletotrichum cercinans. So to overcome these physical and chemical barriers, fungi produces various enzymes, toxins and organic acids and some growth regulators like abscisic acid, gibberellic acid, all these things, enzymes, toxins and growth regulators, I will cover in the coming videos. So you have to remember that to overcome these physical and chemical barriers, fungi used to produce various enzymes, toxins, growth regulators and organic acids. Coming to the third one, through non-cutinized surfaces, how the fungi make their entry or any pathogen make their entry through these non-cutinized surfaces. What are the non-cutinized surfaces present in the host plants? That is like during the seedling stage where the cutin used to be absent, and root hairs which are very sensitive in nature, and buds, flowers and leaves, nectars and stalk ends. Through the seedlings, some of the pathogens like grain smut of jowar, which caused by Sphaerotheca sorghei, and Sphaerotheca cruenta, which used to cause loose smut disease in the jowar, and Sclerospora graminicola, which used to cause downy mildew in jowar and bajra, and Tilacia caris and Tilacia foetida, which used to cause wheat bunt disease. These pathogens make their entry and make their penetration during the seedling stage. And pathogens which make their entry through the root hairs include wilt or disease causing fungi like Fusarium, and club root of cabbage caused by Plasmodiophora brassicae, and root rot of cotton, that is Phaematotrychum omnivorum. And through the buds, pea rust fungi, that is Euromyces piscis, and witch's broom of cherries, that is Staphyna cerassi. These examples are very important, we have to remember. And through the flowers, loose smut of wheat, that is caused by Eustylagonuda triticii, and long smut of jowar, Toliposporium ehrenbergii, and bunt of rice, Neovasia horrida, ergot of rye, that is caused by Claviceps purpurea. These pathogens enter through the flowers. And through the leaves, Basidiospores of white pine blister rust fungus, most of the rust fungus like Cronartium ribicola, which used to cause white pine blister, and they will germinate and grow down into the branches and leaves where Asia used to be produced. And through the nectaries, this is a very important example, especially fire blight of apple, that is Erwinia amelovera bacteria, which used to cause fire blight of apple. It make entry to the nectaries. And other fungi like Penicillium, Telaviopsis paradoxa, which used to cause post-harvest diseases, they will enter through the fruit stalk ends. And through cutinized surfaces where they have come across the cuticle surface, so Cercospora beticola and Alternaria solani and Cercospora personata, these pathogens make their entry through the cuticle. That is Cercospora beticola, which used to cause the disease leaf spot in spinach, and Alternaria solani, which used to cause early blight of solanaceous crops, and groundnut thicca disease caused by Cercospora personata. These pathogens make their penetration through the cuticle. Coming to the third activity, post penetration activities. Okay, after the penetration of pathogen, what it will do in the host cell? First one is invasion and colonization. Colonization means it will colonize among the different cells within the host plant. Infection is a process by which pathogen establish contact with the susceptible cells or tissues of the host, and it derive nutrients from the infected host plant. So it will establish a parasitic relationship between the host cytoplasm and parasite cytoplasm. So there will be a parasite relationship during the invasion and colonization process. During infection, pathogens generally they grow and multiply within the plant tissues, and invasion of plant tissues by that pathogen and growth and reproduction of that pathogen these are the two concurrent stages of disease development. That is, first it has to infect the plant tissue, then it has to grow within that host tissue and reproduce. That is the colonization, means it will multiply within the host system to increase their number. So these are the two concurrent stages in the disease development. Then fungi spread to the all parts of the host organs, either by growing directly through the cells as intracellular mycelium. Intracellular mycelium means it produces the mycelium within the cell, or by growing between the cells, that is intercellular mycelium. If mycelium spreads between the cells and produce the haustoria into the cell, this is intercellular mycelium. Intracellular mycelium means mycelium enters into the cell, that is intracellular mycelium. So these are the two types of mycelium, intercellular mycelium and intracellular mycelium. And during establishment, what the pathogen will do is, pathogen produces different substances like enzymes, toxins, growth hormones and polysaccharides to overcome all the chemical barriers present within the host plant. And this infection caused by microbes, that may be local, or that means local means it involves the only single cell or a few cells or a small area within the infected area. Systemic means it has to spread from the site of infection through the and reproductive structures also it will reach. That is, pathogen spreads and invades most or all susceptible cells and tissues throughout the plant. That is systemic invasion. Example for this systemic and local invasion is the Sclerospora graminicola. It used to cause both local and systemic infection. Then incubation period is the time interval, whatever time interval between the inoculation and appearance of disease symptoms, the time period generally we call it as a incubation period. That is, after inoculation, the time left for the first appearance of symptoms, that is the incubation period. And last stage is the exit of pathogen for its spreading. That is after invasion and colonization of the host. So pathogen come out of the host because it has to maintain continuity of the infection chain and disease cycle, and it has to also escape death due to overcrowding. If overcrowding of the pathogen happen in the host plant, then nutrient depletion automatically it will take place. So that to overcome this overcrowding and to maintain that continuity in the infection chain, it has to exit from the host plant. So once the pathogen exit from the host, then they survive and disseminated to other host and continue that infection cycle. And how these different pathogens they will exit and they will continue the infection cycle, let us see. Coming to the viruses, how whether they cannot exit themselves because they do not have any enzymatic system, so they exit only with the living protoplasm and hence disseminated through their animate vectors like insects, fungi and nematodes. In case of bacteria also they cannot exit themselves, just they will ooze out in the form of slime on the host surface, from where they can be disseminated through water and wind and insects. That is rain splashes, they can disseminate to the other host plants. In case of fungi have the most elaborate system to exit, like most of the plant pathogenic fungi, they grow on the host surface and they will produce repeating spores. That repeating spores what we call as a secondary inoculum. That secondary inoculum is generally asexual spores they used to produce under favorable conditions. So these asexual spores used to be produced in a lot of spores and they will spread to the other host plants. So these spores formed are disseminated through wind, water, soil and seed material, through vegetative propagating material and agricultural implements. This is about the today's lecture. That is what we learned is phenomena of infection and what are the different stages during this infection process, that is pre-penetration activities, penetration activities and post-penetration activities, and how the pathogen will make their entry into the host plant. So thank you for your keen interest in this video, and remember to like and subscribe for more videos. If you like this video, and share with your friends. I wish you great success, health, prosperity and happiness. Thank you one and all.